Hi guys, Miss Francis here to discuss prokaryotic gene regulation. We know that eukaryotes do gene regulation. In other words, they regulate their genes. They turn them on, turn them off. Well, prokaryotes need a way to regulate their genes as well. They need to turn their genes on and turn their genes off in response to any environmental change, which enables them to adjust their metabolism. So, the means by which a prokaryote does this is called an operon. So prokaryotes use operons to regulate their genes, where an operon consists of three parts. You've got the operator. The operator is where the repressor is going to bind. The promoter is where the RNA polymerase is going to bind. There's a TATA region within the promoter, and then you've got the genes of the operon. The genes of the operon are going to code for the protein that the prokaryote wants to make. There are also regulatory genes located upstream, which are going to make a protein known as the repressor. There are two different types of operons. There's the inducible operon and the repressible operon. In this video, we're going to focus on the inducible operon. The classic example of an inducible operon is the lac operon, and this type of operon is normally off, but it can be activated. In other words, it can be turned on. And this type of operon normally breaks down food molecules for energy. So in the example of the lac operon, if lactose is present, we want to turn on the operon so that it can create enzymes to break down that lactose so the prokaryote can use it for energy. So let's annotate this image to help us understand how the lac operon works. So remember, the lac operon is an inducible operon, meaning it is normally in the off position. Well, not normally in the off position. It's normally off, but it can be activated, thus turning the gene on. So let's start by labeling some key components to the lac operon. So first of all, let's start out with this part, which is the section of bacterial DNA that contains a gene that codes for a protein. In particular, the protein that we're looking for are enzymes that are going to break down lactose. We've got an operator, which is where the repressor is going to bind. You've got your TATA box, which is where your RNA polymerase is going to bind, and taken together, these are found within the promoter region. You've also got a regulatory gene located upstream, and what this regulatory gene does is it codes for a protein. In this case, the protein is the repressor. So in this example, we have no lactose. So we don't want to turn this gene on, because if we would turn this gene on, then there'd be nothing for if we turn the gene on, the gene would make these enzymes that break down lactose, but there is no lactose. So therefore, we don't need to make the enzymes that break down lactose. So in that case, we want the repressor to be active. So why do we want the repressor to be active? We want the repressor to be active so that it can bind to the operator, and now RNA polymerase cannot make messenger RNA, therefore the proteins, those enzymes, aren't made. So let's make a flow chart. If my lactose is absent, my repressor is going to bind to my operator. What that causes is a block for RNA polymerase, where the RNA polymerase can't make RNA. Because that RNA isn't made, the proteins, the enzymes that break down lactose in this case, aren't made. And what that means is that the operon, or the gene, is turned off. But now, what if we have the opposite situation? What if lactose is present? If lactose is present, then we would want to turn the operon on, because now we do want to make those enzymes that are going to break down that lactose so the prokaryote can use its products as energy. So we want to inactivate our repressor. So let's once again label all of the different components of our lac operon. So once again, we have a section of bacterial DNA. We have got genes that code for a protein. In this case, it's the enzymes that break down lactose. We've got an operator, which is where the repressor binds. We've got the promoter, which is where the RNA polymerase is going to bind. We also have that regulatory gene upstream, which codes for, which creates our repressor, our protein. Now, I've got lactose present, and what that lactose does is it's going to bind to the repressor, and the repressor is going to undergo a conformational change. It's going to change its shape. So now it can no longer bind to the operator. So there's nothing blocking my RNA polymerase, and now my RNA polymerase can make that messenger RNA, which can then ultimately make the proteins that are going to, in this case, they're enzymes, so they're going to break down that lactose. So let's draw a nice little flow chart for this situation. So I've got lactose present. My lactose is going to bind to the repressor, causing a conformational shape in the repressor. A conformational change in the repressor, meaning the repressor changes shape. And now my repressor cannot bind to the operator. Well, if it can't bind to the operator, there's nothing preventing RNA polymerase from making that messenger RNA. And now I can make the protein. In this case, it's enzymes that are going to break down lactose. So this is an example of an inducible operon where usually the operon is off. However, it can be activated, meaning that it can be turned on. So if we were to summarize this lac operon, which is an example of an inducible operon, if we were to discuss when lactose is not present, where's the repressor? The repressor is bound to the operator region. And then does this enzyme that's going to break down lactose get made? No. The RNA polymerase is blocked. I cannot have my RNA polymerase pass the receptor. So no RNA is going to be made. Therefore, no protein is going to be made. In this case, the protein is the enzyme that breaks down lactose. But we don't need it because lactose is not present. But what if lactose is present? Well, now what we've got is the repressor bound to that lactose or allolactose. So here you've got the repressor bound to that lactose, that otherwise known as lactose. All right, and what that does is inactivates the repressor. So when the repressor tries to bind to the operator, it can't, because it has undergone a conformational change. Its shape has changed. And now my RNA polymerase can read the DNA and can make messenger RNA. Therefore, I can make the enzymes that break down my lactose into lactase, which then can serve as energy for my prokaryote. So where's the repressor? The repressor is bound to lactose. Does the enzyme get made? Yeah, the enzyme gets made because there's nothing blocking RNA polymerase. Is the operon on or off in this condition? The operon is on. I am making that enzyme. In the next video, we're going to talk about the trip operon, which is an example of a repressible operon.